Good day, this is the Professor, CBMD PhD, and today's Wednesday, October 4th, 2023. It is 9:25 and 13 seconds, p.m. Eastern Standard Time, and today's research, Cryo. So let's jump to it here. An advance in Cryo could be a significant boon for research on potential cancer therapies. Team develops key improvement to Nobel Prize winning technology. Date September 28, 2023. Source: University of California. A technology called cryo-electron microscopy enables scientists to see the atomic structure of biological molecules in high resolution, but to date it has been ineffective for imaging small molecules. A team of biochemists devised a solution that makes it possible to hold small protein molecules in place while they are being imaged, which will enable cryo to produce much clearer images of such molecules. The advance is significant because small and medium-sized protein molecules are an area of focus in research on potential new drugs for cancer and other diseases. The scientists engineered a 20 nanometer cube shaped protein structure called a scaffold with rigid tripod-like protrusions that hold the small proteins in place. The scaffold can be digitally removed from the picture when the imaging is being processed, leaving a composite 3D image of just a small protein scientists are analyzing. The small and medium-sized proteins are a hot point for research on potential new drugs that might one day be used to fight some of the most intractable human illnesses. The advance which was tested on a protein that scientists are studying for its possible use in cancer treatments can be customized for almost any small protein. The researchers expect that expanding cryo-EM's imaging capabilities will help them identify specific locations on proteins that they can target for therapeutic purposes. In cryo-EM, scientists use a cryo-electron microscope to send a beam of electrons through frozen samples of material, leaving behind an image of the thousands of molecules, such as proteins, in the sample. The molecules are imaged exactly as they lie in the sample, producing thousands of 2D photographs of the molecule taken from different angles. As computer processing reconciles all those photographs to formulate a correct 3D image, separating the background, grouping images with similar orientations together, and generating a high-resolution 3D image of a single molecule. When it comes to imaging the smallest protein molecules, their tiny size makes it impossible to ascertain their orientations, and the images which result in relatively low resolution images. In previous studies, scientists attempted to solve the problem by attaching small molecules to larger scaffolds, but those experiments demonstrated that if small molecules were attached to flexibility, they would protrude from the scaffold at different angles and orientations, which would still produce blurry images. The scientific investigators and microbiologists say the images are blurry because a computer can't create a distinct composite image when it can't determine the orientations accurately. In the new study, the scaffold created by the scientists had tripod-shaped protrusions that capture the proteins and hold them firmly in place, which yielded the higher resolution images they were aiming for. Attaching the small molecules rigidly to larger scaffolds creates particles that are large enough to be imaged and which all have precisely the same 3D image, and from there, the process works as usual to construct the high-resolution 3D image. The scientific investigators and scientists first tried another shape for the scaffold before landing on the version with tripod-shaped protrusions. At first, they used one stick, pointing outward, and it didn't work as well. The new scaffold has protrusions that point toward each other in triplets, like tripods, that hold the protein firmly. The researchers tested their scaffold by attempting to create images of a protein called KRAS. KRAS encourages cells to proliferate and is involved in about 25% of human cancers. It is of particular interest to pharmaceutical researchers because identifying specific locations on the protein that are related to its cancer-causing abilities can help scientists design drugs that neutralize activity of those locations which can be one path toward treating cancer. Using Cryo and the scaffold they created, the team was able to observe the atomic structure of KRAS attached to a drug molecule that is being studied as part of a potential treatment for lung cancer. Their work proved that the new scaffold Cryo approach can illuminate how drug molecules bind with and inhibit cellular proteins like KRAS could help guide the development of more effective drugs. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.